Welcome today to our talk with Esteban Gurzov, Fixing Dysfunctional Signaling in Type 1 Diabetes, on the right track or lost cause. That's a large and very important question, and I'm really interested to hear what he has to say today. He's actually the group leader in the Signal Transduction Metabolism Laboratory at the ULB in Brussels, and hails from Argentina. He has hit some of the some very interesting laboratories along his training route, including Melbourne University and Monash University, as well as Autonomous University in Madrid, Spain, and for a time he was in Toronto. So I just wanted to give a little bit about a little background for him. Esteban brought to the ULB the particular experience with advanced molecular biology, including complex transgenic models and firsthand experience on the role of protein tyrosine phosphatases in the development of metabolic disorders, including diabetes and liver cancer. He has developed and consistently pursued an innovative research line on mechanisms of insulin-producing beta cell dysfunction and the death in the pancreas and obesity-associated metabolic dysfunction in the liver. The key strength of his laboratory is the combined use of molecular biology, animal models of diabetes, and human samples to translate research from the bench into the clinic. Welcome, Esteban. Thank you so much for joining us, and I'm very interested to hear what you have to say. We have a bunch of people in the room here, and I'm sure they'll have some questions. So if they hold those to the end or drop something in the chat, that'd be great. Take it away.
Thanks a lot, Monica. It's really a pleasure to be in the Sugar Science. It's a fantastic initiative, so really happy to share our research for the community. And indeed, so this is the title of my talk, and I will try to address, I don't know if I'm going to succeed, but at least to give a flavor of what we do in the lab.
Sounds great.
Let's see if I can move. Yeah. So basically, this is our research strategy that we follow. We base our studies in the available screening strategies for type 1 diabetic patients. Clear examples are the genome-wide association studies, but also we look at available data sets and single cell and bulk RNA, seq, and proteomics. We identified some pathways that we know that may be associated to the disease, and we then model the disease with a particular focus on the insulin-producing cells, the beta cells in the pancreas that are associated, obviously, with type 1 diabetes. We work in vitro with human stem cell differentiated beta-like cells, and also with in vivo models where the method that or the model that generally is used are the NOD mice. And we go back and forward between in vitro and in vivo, hoping that we will find a therapeutic strategy that can be then eventually evolved in a clinical trial for the patient. So I will show today some examples of this strategy that we have been following. The main focus of our research and that we base our studies are the protein tyrosine phosphatase. Tyrosine phosphatase are proteins that belong to a family that plays an important role in signal transduction. They counteract the activity of kinases. However, these proteins, they had not been followed as much as kinases, and there are historical reasons. First, that kinases are involved in cancer, so there is a high interest for pharmacological, let's say, inhibitors of kinases. But phosphatase, if we think about it, they are the proteins that do the dephosphorylation. So they turn off the signaling and are as important as kinases for the cells. So I'm focused today on tyrosine phosphatase that we know that are candidate genes for type 1 diabetes, at least some of the members. And they're important not only for type 1 diabetes, but for many other autoimmune disorders. And there are two groups or two main subgroups of tyrosine phosphatase, the receptor phosphatase and the non-receptor phosphatase. And I will focus this talk today in one particular non-receptor phosphatase that is PTP N2. And the reason is because polymorphisms on these tyrosine phosphatase, here in red, have been associated with the development of type 1 diabetes. PTP N2 is expressed in both the immune system, but also and importantly in pancreatic beta cells. And again, so these proteins, they are switch off. So if we think about an autoimmune disorder where the switch off is not there, that may imply that the inflammatory signaling can be enhanced. And basically, this is our main hypothesis, that dysregulated activity of tyrosine phosphatase in the beta cells contribute to the development of type 1 diabetes. So the model that we followed to study the role of PTP N2 was based on the differentiation of stem cells using the protocol established by Doug Melton, Timothy Giffre, and adapted and actually also developed by Timo Tonkowski in Finland. That is the protocol that we got and we follow. Timo is a good collaborator. And we differentiate these cells into beta-like cells. These are human embryonic stem cells. We also work with iPS. And what we observe is, first, that when we differentiate in our laboratory, it's important to validate the protocol that we make insulin positive cells. The percentage of insulin positive cells are around 40 to 60%. And what's interesting to observe that PTP-N2 in the differentiation process is decreasing the expression of this particular phosphatase. And in collaboration with Timo, we use CRISPR-Cas to make knockout of stem cells, of H1 embryonic stem cells. And we differentiate these knockouts into beta-like cells. And what we observed is that there was not measured differences. This is the insulin production, but also in number of insulin positive cells, there were not much difference. So we wanted to study now the signaling of this knockout. So this was done again in collaboration with Timo Tonkowski. Now in the lab, we have generated many other knockouts for different phosphatases, and we are studying the signaling. So we wanted to know what happened when PTP-N2 is not there in these beta-like cells or in beta cells. And we know that PTP-N2 is, as I mentioned in the introduction, phosphatases are switch off of signaling, and they participate in the interferon signaling. Interferons are important pro-inflammatory cytokine for the development of diabetes, type 1, including interferon alpha, and also type 2, including interferon gamma. And both, when they are activated, they phosphorylate the STAT proteins that are transcription factor, and when these proteins are phosphorylated, can form dimers that go into the nucleus and activate gene transcription. So what's the role of PTP-N2? PTP-N2 is a switch off. So what it does is to remove the phosphate for the protein, particularly STAT1, and turn off the signaling. And this is exactly what we observe when we generate beta-like cells without PTP-N2. So we give a pulse of interferon for 1 hour, and then we follow a chase. So these are the control. We give 1 hour, and then after that, we start the chase and look for STAT1 activity. And we observe that STAT1 is activated in the control cells by interferon gamma and also by interferon alpha. However, when PTP-N2 is not there, this activation is significantly increasing in both in type 1 and type 2 interferons. So this experiment was done with differentiated cells, that is a combination of beta-like cells and non-beta-like cells. We are now running single-cell RNA-seq to see what's the response between the beta and non-beta cells differentiated. But we also confirm in a pure human cell line, that is the ENDO-CBH1 cell line, that when we knock down, now using a small interference RNA against PTP-N2, STAT1 is significantly active in both interferon and interferon alpha and interferon gamma. So it's the signaling, the transcription factor is significantly enhanced. So what? Well, when STAT1 with transcription factor is enhanced, activated, our hypothesis was that the signaling was maintained for longer time on the beta cells. And if we think about the patient that has a polymorphism that inactivate the the function of PTPN2. That means that when those beta cells are exposed to a cytokine environment, they will maintain the exposure or the signaling, this inflammatory signaling, active for a longer time. So they will not be able to switch off the pathway. And to test this hypothesis, what we did is, again, to give a pulse of the cytokines for 1 hour, and then we remove the cytokines and we follow the chase, and we perform RNA-seq analysis at 24 hours. So 24 hours after the cells were exposed to the cytokines. And first we confirmed that the knockdown of PTPN2 actually knocked it down and reduced the expression of PTPN2 in all conditions. And what we observed is the inflammatory cytokine was enhanced and that our hypothesis was correct. So when the beta cells do not have PTPN2, 24 hours later, the signaling is significantly higher in both interferon alpha and interferon gamma. So to give an idea, only 500 genes were still active in the control cells, where more than 1,400 genes, almost 3 times more genes, were expressed 24 hours when PTPN2 was not there. And something that was also interesting that we observed is that the ER stress response was significantly higher when PTPN2 was not there and the cells were exposed, the beta cells were exposed to the inflammatory cytokine, suggesting that maybe PTPN2 was also able to modulate the ER stress response. And to test this hypothesis, what we did is to knock it down, again, PTPN2 and ENDO-CBH1 cells with 2 different RNAs, and expose the cells to an ER stressor, or to actually 2 ER stressors, tapsigargin and CPA. And what we observe is that the cells that have deficient PTPN2 activity is the cell death, it was higher, making the cells more susceptible to ER stress. And not only the cell death was higher when PTPN2 was knocked down, but also obviously the signaling was exacerbated. So all this was done by qPCR, and we observed actually that all the pathways of the ER stress response was enhanced. This was qPCR. We could confirm the same by Western blot. I'm showing 2 of them, the proteins that are activated by the ER stress and that are significantly enhanced when PTPN2 was knocked down. And this in endo-CBH1 cells. In the human beta-like cells, knockout for PTPN2, we observed the same. So BIP, that is an important chaperone activated by ER stress, was significantly higher. And importantly, in these first human islets, when we knocked down PTPN2, these islets were provided by Piero Marchetti from Italy. When we knocked down PTPN2 and we exposed the cells to the ER stress, again, the ER stress response was higher. So not having PTPN2 make the beta cells more susceptible to ER stress and to inflammatory cytokine. What happens if we enhance the expression of PTPN2? For this, we used Adenovirus that express either or the 45 kD isoform or the 48 kD isoform. And the difference is that the 45 isoform shuttle between the nucleus and the cytoplasm, and the 48 is located at the ER. So we tested what happened when we overexpress these proteins in the beta cells, and I'm not going to show all the data with the 45, but only with the 48 that is located at the ER. And what we observe is that ER stress response is decreased when we expose the cells to tapsigargin, and also there is a partial prevention of cell death. And what we observe with the 45 kD isoform that shuttles between the nucleus and the cytoplasm, that is important for the inflammatory cytokine signaling induced by interferon alpha and interferon gamma. So to summarize this part that's been recently published in Diabetes, we observed that PTPN2 the deficiency enhanced the pro-inflammatory gene expression in beta cells, that there are 2 isoforms of PTPN2, one that shuttles between the nucleus and the cytoplasm, and the other that is located at the ER that control the ER stress response. And I'm not going to show here the data, but we compare our RNA-seq data set with the profile of beta cells from type 1 diabetes that was published by Russell et al. Some years ago, and what we observe using this RIHO, that there is a correlation between the signaling of the inflammatory modulation when PTPN2 is not there and disposed of interferon with the signaling associated with beta cells from type 1 diabetes. I take this with some concern because that's only gene expression, but it seems that actually the inflammatory response when PTPN2 is not there correlates with type 1 diabetes. So now the question, this was all in vitro. The logical question is, can we increase PTPN2 activity in beta cells or islets in vivo and have an impact on autoimmune diabetes? And to address this, we use two models and two mechanisms. First, we use a low dose of anti-CD3 with interleukin-1 receptor antagonist. This was done in collaboration with Connie Gissemans from the KUL and with Wei Wu from the Netherlands for the proteomics. And the idea was to reverse diabetes in the recently established diabetic NOD mice and to see what proteins are modulated in the mice that being cured. So we give the dose for 5 days, and first we confirm that this treatment with the low dose of anti-CD3, now is trendy, and interleukin-1 receptor antagonist actually reverse diabetes in the NOD mice. This was published, so this was not new. And compare with a low dose, again, I emphasize that, it's a low dose of anti-CD3 that did not reverse diabetes. So we look at what happened in the proteins that are modulated in the pancreas. I'm not going to enter into all the pathways that we observe, but related to phosphatase, we observe a small increase in the total phosphatase, although not significant. And not surprisingly, the phosphatase that being highly activated in the cure mice was PTP N2 or highly expressed. And we also observe a small change in PTPN13 that is expressed in the immune system. And we are currently studying what's the role of these phosphatase. The other phosphatases that we detect were not different. And this was proteomics done in the whole pancreas, that obviously the beta cells are a minority. So to confirm that PTPN2 is expressed at least in some of the beta cells, what we did is immunostaining, and we correlate the expression of PTPN2 with insulin, meaning that at least in correlation, the beta cells that have been protected, they have high level of PTPN2.
I'm just going to read out a chat that just came through like 2 minutes ago, so maybe 2 slides back. Do you know which PTP N2 polymorphisms correlate with the full KO, full knockout, and to what extent?
So in the knockout, we knock out all PTPs, both isoform 45 and 48. So what we know is that indeed that not ourselves, but it's now in the field that the 45 kb lack a C-terminal region that allow them not to be allocated in the ER compared with the 48 kb that in the carboxy terminal, they have a region that locate the protein into the ER. And what we think is that that location, actually, what we demonstrate in the paper is important to interact with chaperones and protein at the ER. And this is how actually ER stress is controlled by PTPN2. I don't know if that answered the question, but so
well,
when we knock it down, we did not knock it out one or the other. So we knock out the complete PTPN2.
Okay.
And the
I was asking about the polymorphism, so the actual single nucleotide mutation, basically, how not the form, not the isoform.
Yeah, so the polymorphisms, there are several polymorphisms associated with PTPN2. Some they are located in the intron region, and what was done actually, the studies that have been done to see what is the effect of this polymorphism is a loss of function, and is in response mainly to the 45 kb, so meaning that is important for the dephosphorylation of proteins, particular STAT proteins. So the ER was something that actually we found, and there were other laboratories looking at the role also of these ER isoform in the context of obesity, but in type 1 diabetes, we are actually the only one who found that there is a role in the ER, and it's mainly by the 48 kb. But the polymorphism, we don't know whether it's associated actually, or they are impacting on at the role of PTP-N2 at the ER. That's something that need to be addressed. So continue with the second model that we have to activate the phosphatase. So before we look at the expression of PTP-N2, however, phosphatase and tyrosine phosphatase can be inactivated by a second mechanism, that is through oxidative stress. And we know that the inflammation, there is oxidative stress, and why phosphatase can be inactivated by ROS, or by reactive oxygen species, is because of the structure of the proteins. All the tyrosine phosphatase, they have a conserved catalytic site. This is the structure of PTP-N1, but it's the same in all the family. They have a cysteine and arginine, and the cysteine is critical for the nucleophilic attack. So to do the job, the phosphatase, they need the cysteine and to provide a nucleophilic attack over the phosphate of the substrate. And this characteristic of the catalytic site made them actually highly propense to oxidation and inactivation by oxidative stress. So phosphatase can be inactivated by ROS production, and we know that in inflammation we have oxidative stress. We observed that in the context of obesity, but when I started my laboratory at St. Vincent's, I had a very talented PhD student, and he studied the role of oxidation of phosphatase in NOD mice. So what you can see in this Western blot, each line corresponds to a pancreas of one mouse. These are the controls that do not develop diabetes. These are the NOD mice that do develop diabetes. Each line corresponds to a phosphatase, and you can see that in the mice that develop diabetes, there is oxidation of phosphatase. And we show in the paper actually that one of the phosphatases that get oxidized and inactive due to this inflammatory context is PTP-N2. So we wanted, So we wanted to actually reverse this oxidation and this inactivation of phosphatase. We tested different antioxidants. Antioxidants, they have many caveats and concerns, actually. So what we wanted is to find a more physiological way to modulate the oxidative stress in the pancreas. And for that, we know that actually short-chain fatty acids can reduce oxidative stress. And interestingly, and that was at the time where I was in Australia, I started collaborating with Eliana Mariño, that was at Monash as well. And she just recently published at that time a paper showing that hams, so a diet that consists in the high amylose maize starch, that's the backbone, that being acetylated or butylated, actually prevent the development of diabetes in NOD mice. And there is an effect of the diets when they have either acetate or butyrate compared with the controls. And since we knew that acetate and butyrate actually prevent or decrease oxidative stress, we wanted to know what happened with the oxidation of the phosphatase in the pancreas at the early stages before diabetes development. And we observe indeed that acetate and butyrate, as expected, decrease the level of oxidation of the phosphatase, meaning inactivation of the phosphatase, including PTP-N2. So we shown that short-chain fatty acids prevent ROS formation and inactivation of tyrosine phosphatase in autoimmune diabetes. So then the effect was, what are the effect and signaling altered by short-chain fatty acids? So oxidation of phosphatase is prevented. What happened with the signaling in the autoimmune diabetes? And before to show the data that we have, a small remind what short-chain fatty acids are. Short-chain fatty acids are produced by the dietary fiber, are produced by the gut microbiota when we eat fiber. And the main example of short-chain fatty acids are acetate, butyrate, and propionate, and they have an important role in the organisms. They are produced by the bacteria, for our own bacteria, and they maintain a healthy intestinal mucosal barrier. they enhance the immune function, decrease oxidative stress, and prevent inflammation, all things that we want in type 1 diabetes. And also they have an important endocrine role that reduce appetite, increase glucose uptake, and decrease insulin resistance. So what we did in the lab was to reproduce what Eliana have done in Australia, now here in Belgium, again with Connie Gisman from the KUL. So we give to the NOD in her colony the diets. We observe actually again a reduction in the diabetes development, and we wanted to know what happened with the signaling at early stage in the islets. So for that, we use the HAMS-AB that produce acetate and butyrate, and the HAMS diet as control. We harvest the islet and we run a single cell RNA-seq to understand what are the signaling modulated by this diet in the islet. Interestingly, in the single cell, we observe in both, it has HAMS-AB and HAMS, the presence of both endocrine cells and immune cells. And there were slight enhance of endocrine cells, but not much difference between the HAMS-AB that protects from diabetes versus the HAMS diet. And I can go through all the different populations, but I will focus now only on the endocrine cells to understand what's the signaling affected. And what we observe is in line with what we found, that PTP N2 actually modulates STAT1, that STAT1 is decreased in the HAMS-AB or increased in the HAMS in the beta cells from the mice that develop diabetes, that many stress genes were activated in the beta cells that from the mice that develop diabetes, and the identity genes of the beta cells were maintained in the HAMS-AB, the diet that protects, that produces acetate and butyrate. And it was known and published by others, but we reproduce the results that acetate and butyrate actually increase beta cell gene expression identity. And we did the same experiments that were previously published in the INS1 beta cell line. But we are now also studying this effect in stem cell derived beta-like cells, because it can be one of the mechanisms how we can improve actually the differentiation of the stem cells. And this is something that we are testing now. And regarding other endocrine population, and something that was we found very interesting between the control mice that develop diabetes and the ones that are prevented from diabetes, is that in the population of the alpha cells and the delta cells, we observe a cluster that here is represented by the cluster 1 in the HAMS-AB, that were a minority in the HAMS-AB, and a cluster 2 in the delta cells that is pretty much inexistent in the HAMS-AB. And this cluster of cells corresponds to polyhormonal cells that start expressing genes from other endocrine cells. We think that this might be the differentiation or loss of cell identity. And this was not new. It was observed actually in NOD mice before, but more importantly in the samples from type 1 diabetes. So these polyhormonal cells, what are the role of these polyhormonal cells? Still, we do not know whether they're functional or not, probably not. But we are trying now to identify, and using again the stem cell derived beta-like cell and differentiation process to see what are the effect of the HAMS, particularly the acetate and butyrate on this differentiation process. So we observed that the diet that protects from diabetes in NOD increase beta cell function, in functional genes, reduce the stress response, and preserve the identity of the endocrine cells. So to summarize this part, what we observe actually is that when we combine anti-CD3 low dose with interleukin-1 receptor antagonist, we increase PTP N2 expression in beta cells and islet from NOD mice. You may ask if this cause or consequence, and this is something that we are also eager to study using knockout cells and transplanted actually knockout cells, humans, beta cells and to see what happened with those differentiated cells that do not have PTPN2 when we give this treatment. And actually also would be nice to have the knockouts in the NOD for PTPN2 to find whether it's a cause or a consequence. But we know that PTPN2, deficient PTPN2, beta cells that are more susceptible to inflammatory signaling. They signal the inflammation for longer time, make the cells more attractive for the immune cells. In terms of signaling, we observed that also enhanced chemokine suppression, enhanced the MHC proteins, meaning that those beta cells, they become more attractive for autoimmune attack. And also PTPN2 make the cells more susceptible to ER stress. And when we overexpress, at least we ameliorate this dysfunctional signaling. How we can increase? Well, we observed that the anti-C3 interleukin-1 receptor antagonist increased the expression of PTPN2. And I did not show the data because it was published, but mainly we think that is through Tregs effect and in hands of interleukin-10. We observed that interleukin-10 actually enhanced PTPN2 expression in the beta cells. And the other mechanism of inactivation of phosphatase is through oxidative stress mediated by the inflammatory process and the HAMS-AB. Obviously, these two protective systems, they do much more than this, but related to phosphatase activity, they decrease HAMS-AB diet, decrease oxidative stress, and improve beta cell function. So the HAMS-AB have been published before that also have an effect in the Tregs, an important effect in the immune system, and that was published by Eliana Marino in Nature Immunology. Our studies now with single cells that have not been published yet also highlights that the diet can improve the functionality of the beta cells and the cells in the islets, and supports the development of clinical trials using the diet in patients. And indeed, I was part of a clinical trial using this diet. This was funded by the JDRF, and the study was designed mainly by Liana Marino and Emma Hamilton-Williams. There is a fantastic talk by Emma in the Sugar Science presenting this clinical trial using this HAMS-AB diet. And also Kirsten and Sonia, where Kirsten was involved in the patient recruitment and the study with the patient, and Sonia in the bioinformatic analysis. So I have a minor contribution, but what's important to show that actually this diet, they have a positive effect on people with established type 1 diabetes and patients with established type 1 diabetes. I'm not going to describe much of the clinical trial because, again, it was presented some weeks ago by Emma. So it's a 6-week, it's a pilot study. The patients that received the diet for 6 weeks and had a 12 weeks follow-up. And the conclusion, if you haven't seen Emma's talk, spoiler alert, the results were really positive in this pilot study. It was safe and tolerable in the patient with established type 1 diabetes, induced a change in the gut microbiota that was important, that returned to baseline after we finished the study. And there was actually correlation with plasma butyrate and fecal acetate with glucose control. This was a pilot clinical study. So now a more developed study should be done with placebo and with people at risk of development type 1 diabetes. So the perspective is that, and what I would like to briefly discuss is that we know some pathways that can contribute to the development of diabetes, but there is a high heterogeneity in the patient, and not many of us actually talk about it. We discussed T1D as one disease, but I think there are different endotypes, and it's definitely not the same of patient that develop diabetes at early age with someone that developed diabetes at 30, 40 years old. we need to start conceiving the disease as a different, let's say, subtypes of disease, and we don't know what signaling can affect in one patient or the other. And something that is important, that there is a high heterogeneity also in the same pancreas in the same patient, not only between patients, but in the same patient in the pancreas. There are islets that are completely protected from the inflammation, from the insulitis, where other islets are completely destroyed. And we look, and when we see papers, we see, including our own papers, we see 2, 3, 30 islets, but in a pancreas it have between 1 million to 10 million islets. All the heterogeneity there, why these some islets are protected and other are not, is something that we do not really know. So we need to start thinking about the study the whole pancreas in 2D, or even better in 3D. And for this, I refer a collaborator, Ulf Algren from Sweden, who actually developed a 3D structure and was able to image the 3D whole human pancreas. And I know that he's also working now with type 1 diabetes.
Esteban, who was that again? Could you repeat that name?
Ulf Algren from Sweden.
Oh, Ulf, yeah.
From UMEA.
Yeah. And I think Guy Rutter and, Richard Benninger are also kind of addressing that idea of the endotypes, of cells within one pancreas, human pancreas, like what, why are they, differentially attacked?
And what we are looking, because that's what we have, is the samples from donors. That is very valued material, but that's the crime scene, but of the beta cells. But the crime was probably planned many years before by the immune cells, and we do not have the complete picture. So we have some clues of a small portion of a pancreas, and we make big studies based on that. And what can be relevant for some patients may not be relevant for the others. So we really need to understand more the disease. And I think it's actually important times and interesting times now with the development of technology. And this is the second point, that we need to some people do single cell RNA-seq, others do other type of transcriptomics. Some people work in metabolomics or immune cells. So I think we need a more multi-omic kind of studies involving different techniques. Spatial transcriptomic is there. The pancreas is quite complex because of all the digestive enzymes. However, I think we will be able to do spatial transcriptomics soon, combined with single nuclear RNA-seq, for example, in human pancreas, and proteomics, and even single cell metabolomics, that technique that are there. And we need to start thinking in this way to analyze how the different genes and actually how different pathways are affected. And the important thing is to consider that's why some beta cells are protected and why others are not in the same pancreas.
Yeah, it appears, in speaking to a number of scientists, that hopefully the understanding the etiology, the prodrome, is going to yield more information to the knowledge base about, how someone progresses to type 1 diabetes. And I would also bring up the fact that scientists have said, well, we don't want to talk about endotypes, even though we know that they are there, because it's not a good business case for a farmer and others to get involved. But if you think about breast cancer, originally it was one size fits all for breast cancer. And then ultimately now it's, very personalized medicine in its treatment. So I don't understand, it seems to me that the type 1 diabetes could go that same route eventually.
It's the elephant in the room. We know that patients, they develop different type 1 diabetes, but no one talk much about that. And what you say, it's important. And Matthias Bonharath, he was
Mhm.
having this idea of combination of therapies, and I think it's important, this combination of therapy in which patients actually may be effective and when and how. And the last point is, as every single disease, the early we intervene, the better. And type 1 diabetes is not an exception.
No,
for
I
that,
agree. I have a question about this PTP N2 polymorphism. I mean, if it can it be detected at sort of an early well baby check? And then for those individuals, you know, they could undergo further test of their microbiome to see whether or not they're, you know, SCFA producing bacteria are at healthy levels.
yes,
Or even modify their diet at that point if they can understand that, okay, this is someone who has, you want to call it a biomarker for PTP N2,
yeah,
they could be addressed. Is anyone doing that? Like trial and
not
error?
really, and that's why I emphasize the early diagnostic screening to know not only autoantibodies. I think the genetic composition. Obviously, SNP is a susceptibility, let's say, characteristic. Does not mean that the person will develop the disease,
Yeah.
but at least to have the person under screen and based on those possible SNPs associated, and if there is a family history of T1D, I think it's important to know, and for the future to try to identify the patients that may go to one treatment or the other, and to know whether they will be at risk. For now, I mean, the screening is autoantibodies, and even we are not there yet. So I think that the pilot screening now in Australia, also run by Christy, it's important to see which method is more susceptible, and in Australia they are thinking already to go through a full population screening in the future. And I think that's important. But again, we do not know many pieces of the puzzle, and we are treating T1D as a global or a common disease that we know that is not the case, and we really need to study more the signaling to understand how this disease happen and what is the difference and heterogeneity between the patient.
Yeah, I mean, even as early as, you know, this year, there was another paper that came out from the HPAC consortium and others. Mark Atkinson was involved, Al Powers at Vanderbilt, with alpha cell dysfunction islets from the non-diabetic GAD autoantibody positive people. So that, you know, and the BCM beta cell mass was still fine, but the alpha cells were going offline first. So that it just suggests more and more that the endotypes are alive and kicking. So,
yeah, and indeed, some people think that alpha cells are not destroyed, but obviously, I mean, there is an effect of the immune cells there as well. It's true that alpha cells are somehow more resistant, but they are not directly the target of the immune cell.
yeah.
So, and again, it could be that from some patients is one way, and for the others is the other. And I'm also intriguing about the heterogeneity in the pancreas of the same person. So why some islets are destroyed and other not. Sometimes maybe because they increased PD-L1, so there is an inflammatory role there, but it's really unclear. And this is something that we need to solve in the future.
Yeah,
If
we
you
need the
allow me-
best and the brightest postdocs to join the fight. I'm opening it up to the rest. Any other questions? You feel free to unmute yourself or just drop something in the chat while he notes all his collaborators.
Yeah, I would like to thank the people that actually did most of the work in my lab. Bernat Elvira, a talented postdoc that now moved to the private sector, and Valerie, that is doing her PhD, super talented PhD student. Many people in the lab also involved, the collaborators, the participants of the trial, the funding, and if students or postdoc are watching the video in YouTube and they want to apply, there are opportunities in my lab, so please write an email. With that, I really thank you for the invitation.
Thank you very much. I appreciate you going a little over. It was a fascinating talk and really, really great to hear what's going on in your laboratory. Thank you again, and we'll be in touch again soon. Have a great evening.
Thanks so much, Monica.
Bye-bye.
Bye-bye.